good information in the introduction, so I just want to hit some of the highlights of that introduction. So first of all, our focus is going to be on bacterial flagella. So bacterial flagella consist of three main parts, all of which are made out of proteins. So the three main parts are the basal body, the hook, and the filament. So the basal body is the portion of the filament that's anchored in the cytoplasmic membrane in the cell wall. Okay. The hook extends from the basal body and connects the basal body with the filament. The filament is what we would see, say we had a flagellar stain using a light microscope. We see these long thread-like extensions. Those represents the filaments. And you all remember that bacterial flagella, they rotate like a boat propeller, and that's how they propel the bacterium through its environment. We briefly mentioned, I think in lecture, that bacteria have chemical receptors in their cytoplasmic membranes, so they can use chemical concentration gradients to guide their movement. That's called chemotaxis. Some microbes that carry out photosynthesis have light detectors or photodetectors, so they can move to different areas of the environment with different degrees of light. That's called phototaxis. Now, the bacteria can be classified depending on if they have flagella, and if they have flagella, their arrangements. And this actually turns out to be an important part of identifying unknown bacteria. So what we next want to do is we want to go over some flagellar arrangements and give you the conventional terminology, and of course we want you to know the conventional terminology on the lab practical. So once again, Professor Holland has downloaded some beautiful, in this case, photomicrographs of different bacteria with different flagellar arrangements. So we'll cartoon the arrangements, we'll write down the specific terms for those arrangements, and then we'll see if we can find some photomicrographs that match. Okay, so here's my little individual bacterial cells with different flagellar arrangements. Now, a lot of bacteria lack flagellae. They don't have the genes to make flagellae. So they're simply called atrichous. Trichos referring to a hair or a whip. So A is without. So atrichous is a bacterium that lacks flagella. Now, some bacteria will have a single flagellum at one pole. That's called monotrichous. Mono means one. Monotrichous. And in these cartoons, this middle bacterium is monotrichous, single beautiful flagellum. Now, some bacteria will have a flagellum at either end, and that's called amphitrichous. Amphitrichous. We don't have a photomicrograph of an example. Some bacteria have a tuft or a little group of flagellae at one end, and that's called lophotrichous, for a tuft of flagella. And on this example, the third bacterium down below is lophotrichous. And then my favorite is this last arrangement where the bacteria have flagella covering the entire surface. This is called peritrichous. Peritrichous. And in Professor Holland's examples, it's this top bacterium, peritrichous. Okay. So again, flagellar arrangements can help in classifying unknown bacteria. Okay. Now, in addition, if we're talking about classifying bacteria, we can use antibodies that humans and animals make against the flagella. Okay, so we're going to back up here and talk about flagellar antigens. So first of all, we want to know, what is an antigen? An antigen is any substance that can trigger an immune response. And one of the classic immune responses we like to discuss are antibody productions. Okay, so it turns out that since flagella are made out of proteins, proteins are excellent antigens. If we were to inoculate, say, some laboratory animal with flagellar antigens, the animal would make really high concentrations of antibodies against those antigens. Okay, so historically, yes, sir?
What's the difference between an antigen and a pathogen?
Okay, so a pathogen A pathogen is an organism or microbial agent that causes disease. So a pathogen is an organism or an agent that can cause disease. Invade another organism.
Doesn't it cause an immune response like an antigen does?
Causes disease. Okay, and you're absolutely right. Most pathogens are also antigenic in that they'll trigger an immune response. And not to make you crazy, we'll go into this in lecture, but let's say we had a bacterium here. And let's say this is an invasive bacterium. And let's say it has a flagellum. And then let's say it also has a variety of other maybe surface molecules. I'll just put different colors. Okay, so this is a pathogen. And it is an antigen. Okay? It will trigger an immune response. And what we're going to see is that different types of antibodies will be produced against different parts of the pathogen. So we'll have antibodies against the flagellum. We'll have another set of antibodies against this green surface molecule. We'd have another set of antibodies against this kind of oval-shaped molecule. So a pathogen is an organism or agent that can cause disease. Most pathogens are antigenic in that they will trigger disease, but we're going to encounter one pathogen that's not antigenic. And that, I should write this down. So most pathogens are antigenic, meaning they'll trigger. Most pathogens are antigenic, meaning they'll trigger an immune response. But there's some wild exceptions, and one of them are the disease-causing prions. We're going to discover disease-causing prions are not antigenic. They don't trigger an immune response. So most pathogens are antigenic, but not all the time. Okay. So sorry, you guys. I know, and the whole immunology is such a cool field. We could start talking about it now and never stop. So you better stop me right now. Just say, okay, Karen, stop. Okay. All right. So we're going to go back to the flagellar antigens. Okay, just like this little bacterium here, it has a flagellum. So the flagellum is made out of proteins, triggers a really strong antibody response. Now, what's really cool is it turns out that different bacteria can use different types of proteins to make their flagellum. So there's different flavors of flagellar proteins. And those different types or flavors, and you can think I'm thinking about food, will trigger production of different types of antibodies. So we can use those antibodies directed against specific types of antigens to help us identify unknown bacteria down to the strain of the bacterium. Okay, so this process by which we use antibodies to identify unknown microbes is called serotyping. Okay, so in serotyping, we use antibodies to help identify specific strains of microbes. Okay, so that's where we're heading with this. It turns out that amongst one of our really familiar bacteria, good old E. coli. Okay, well, we talk about E. coli as if it's a single species, which it is, but there are hundreds and hundreds of different genetic strains of E. coli. Some of them are beneficial. For example, the E. coli living in your intestines right now, they're beneficial. They actually help protect you against invading pathogens by binding to receptor sites. They compete for nutrients. Some of them make vitamins for us. So there's many beneficial strains of E. coli, but there are also some very harmful strains of E. coli, those that carry genes to make toxins. So as an example of some toxin-producing strains, one that you guys have probably heard about is the strain called E. coli 0157H7. Okay, and so we want to develop, what do these numbers mean? Okay, so we see here this H. That H is the H antigen. Those are the flagellar antigens. And I want to give you just a little bit of history of why these flagellar antigens are called the H antigen. So it goes back in history to German microbiologists who were first studying bacterial structure. And what the German microbiologists discovered is that some bacteria, when you inoculated an agar plate and incubated them, they appeared to swarm over the surface of the plate. And again, this is from Professor Holland. And so this swarm-like growth on the surface of the plate, it creates these like waves of growth. Okay, this is classical for the bacterium Proteus that you guys inoculated into your Motility agar deeps. Proteus have Paratrichus flagella everywhere. So if there's a little bit of moisture on the surface of your plate, they swarm, they just swim in these waves over the surface of the plate. And as a consequence, when you pull your plates out of the incubator and you look at the surface of the plate, you see this kind of cloudy opacity on the surface of the agar plate. And what it reminded the German microbiologists, it reminded them of when you go in the bathroom and you breathe on the mirror and the mirror clouds up. It had that same appearance on the surface of the plate. Well, the German word for breath is Hauch, H-A-U-C-H. Okay, so they wanted to figure out what permitted the bacteria to swarm over the surface of the plate, giving this breath-like or Hauch-like appearance to the surface of the agar plate. And they soon identified it was the presence of flagella that permitted the bacteria to swarm over the plate. So historically then, the Flagellar Antigens have been named the H antigen, the Hauch antigens, right? Okay, so looking at this, Hauch, looking at this, German words are just great, Hauch. Yeah, okay, so if we look at this strain of E. coli, and this is the serotype, it's strain identified by the kinds of antibodies it will stimulate production of. We look here, it says H7. This tells us there's at least 7 different types of flagellar antigens, right? And this particular toxin-producing strain of E. coli makes the 7th type of flagellar antigen, the 7th type of flagellar protein. Okay, so that's how serotyping can be used to track these disease-causing strains of bacteria, especially E. coli. Now, the other one, and it doesn't have to do with flagella, you guys, but just so we finish the story, you'll see here in the serotype name, there's an O there, and we just want to finish the story. So O stands for ohne Hauch, and ohne is without. So ohne Hauch. So when the microbiologists identified there was another component of bacteria that could trigger antibody production, but what they discovered was this component had nothing to do with motility. It had nothing to do with the ability of the microbes to form this breath-like appearance on agar plates. So they said it's something that doesn't have to do with this breath-like appearance, something that doesn't have to do with motility. So they called it ohne Hauch, and we call it now the O antigen. And the O antigen, if we were to identify, or if I were to ask you what is the O antigen, it's the polysaccharide portion of LPS. What does LPS stand for, you guys? I know this is going back a bit. Lipopolysaccharide. Good. Where do we find lipopolysaccharide? In which group of microbes? Good. Excellent, you guys. So LPS, you guys remember, is found in the gram-negative cell wall, the gram-negative bacterial cell wall. And which specific part of the gram-negative cell wall do we find the LPS in? Good, I heard it, you guys. The LPS is found in the outer membrane, right? And what's another interesting factoid about LPS? It's called endotoxin, right? Yeah, endotoxin. Good, okay. So we want to remember LPS stands for lipo, the lipid A portion, the lipid portion, lipopolysaccharide. So saccharide, a change of sugar residues. So it's this polysaccharide portion that is antigenic, that will trigger specific types of antibodies to be produced. So it's this polysaccharide portion then that is the O antigen. Okay, and there'll be different types of sugars linked together, and that will trigger different types of antibodies to be produced. Okay, so again, you guys, if we go back to this toxin-producing strain of E. coli, and we look at the O antigen number, we see there's at least 157 different O antigens out there amongst these gram-negative bacteria. And this particular toxin-producing strain of E. coli has 157th variety. Okay, so these O antigens and H antigens are really important in tracking disease outbreaks. This E. coli 0157H7, it's shed in cattle feces, in human feces, animal feces. So wherever the feces go, the toxin-producing E. coli goes. So we're really worried about fecal contamination of food and drinking water as a source of these E. coli outbreaks. And so when people start presenting in emergency rooms with bloody diarrhea and kidney failure, which is a common sequelae for these infections, it's really important for the public health microbiologists to figure out where are they getting sick. What is the source? Is it contaminated water? Is it contaminated food? And so they'll go back and start screening food for the presence of this specific type of E. coli. Now we want to remember that the E. coli growing in our intestinal tract doesn't make the sugar, this is the toxin this guy produces is called chemotaxin. Okay. The E. coli growing in our intestine, they're of a different serotype. They don't produce the chemotaxin, right? So we wouldn't worry if we found maybe, I don't know, this would be pretty gross. I mean, water that had another type of E. coli growing in it and didn't have that chemotaxin producing strain. Yep. And then just one last tidbit, you guys. Last May in Europe, they were totally taken off guard because people started presenting with bloody diarrhea and some of them started going into kidney failure. And they isolated an unusual strain of E. coli called E. coli 0104H4. So this is kind of a new emergent chemotaxin producing E. coli that now hospitals here in the United States will have to start screening for. So again, we see they can track these different strains of E. coli based on their O antigens and their H antigens. Okay, well that's a little blurb on the O and H antigens. So the last thing we want to hit is our experiment trying to determine whether two different bacteria were motile or not. And unfortunately our Proteus didn't read the lab manual. And so we ended up with some false negative results on our motility auger deeps. But let's just review it really quick. Okay, so you'll recall with your motility auger deeps, you took your needles and you stabbed straight down and straight back up. And so what you did is you deposited your bacteria along the stab line, right? So then you incubated them. Now the idea is if your bacteria lacked flagella or was not motile, they could only grow along the stab line. Now that's what we were predicting for the Micrococcus that you inoculated. Micrococcus lack flagella. So they're atricous. So we would predict that they would be not motile. So non-motile, they can't move. So when you hold your Micrococcus tubes up to the light, we would have predicted you only see opacity along the stab line where the Micrococcus were growing. They don't have the ability to move away from the stab line. Now, what we were hoping you would see, but the Proteus in most cases when we checked didn't cooperate, what we were hoping was that the Proteus, since it has paratrichous flagella, we were hoping that it would show a lot of motility, a lot of growth away from the stab line. So what we were hoping you would see, and it didn't happen, so, and in most of your tubes unfortunately, for Proteus, which is, it has flagella, it has the paratrichous flagella, and therefore we were hoping it would be very motile. What we were hoping you would see is that the cells were growing away from the stab line into the surrounding auger. And very often, if it works well, what you see is a lot of movement away from the stab line up top where there's more oxygen present, where they can carry out aerobic respiration, make 38 ATP per glucose. And then often what you see is less motility down towards the anaerobic gut, where they have to switch over to either anaerobic respiration or fermentation. But sadly, our Proteus didn't exhibit much motility, and it might have been my fault for us going at 37. Maybe they would have been happier at 30 degrees Celsius, right? So I do want to apologize that that didn't work for us. But and I totally understand why you guys are concerned. The follow-up question would be, well, since our tubes didn't work that great, would you still have them on the lab practical? And I think what we'll do is we'll use a probably use a photo of motility agar that has an indicator in it that turns color where the bacteria are growing. Our motility agar deeps are a little bit hard to interpret because you're basically looking for the shadow of where the cells are growing. And to me, it's often really hard to distinguish that increased opacity in the agar. So let me, what I'm going to recommend to you guys, take a look at your photo atlas, look under motility agar, and okay, so in the old photo atlas, it's on page 67, 68, I think. Oh, here it is. Okay, on page 68. This is the type of motility media that's really nice to use, really nice to interpret. And in this motility media, it's the one up top here, guys, so you can see the little red. This medium has an indicator in it that turns red wherever the bacteria are growing. And so then it's not hard to interpret. You're not looking for like, is that a little bit more, opaque than the surrounding medium? So in this particular example, we can see one bacterium was inoculated along the stab line. They were not motile, so they could only grow along the stab line. See that? Okay, so that would be evidence of a bacterium that's not motile. In contrast, in this other tube on your left, this bacterium, and this looks like a Proteus, this bacterium was very motile. When it was inoculated to the stab line, it started growing. It started using up the nutrients along the stab line. Metabolic waste products accumulated. So the bacteria started moving away from the stab line into the fresh agar. And as a result, we see that indicator turning red almost throughout the entire agar tube. Okay, so on the lab practical, I'd probably include like a photograph of motility agar that has that indicator incorporated in it. So it will be really easy to interpret. Okay? And again, I'm so sorry that our Proteus didn't read the lab manual. And like I said, it might have been, maybe they weren't happy at 37, so that might have been my mistake. Okay, so you guys all right with motility? Okay, all right. Now, we'll move on then to Chapter 11, Aseptic Transfer of Bacteria.